Good morning, ladies and gentlemen, and welcome to Alector's conference call and webcast highlighting its progranulin franchise and Alector brain carrier programs. As a reminder, this conference call is being recorded. Currently, all participants are in a listen-only mode. There will be a question and answer session at the end of this call, and to participate, simply press star 11 on your telephone and press star 11 again to remove yourself. Now, I would like to turn the call over to Katie Hogan, Senior Director of Corporate Communications and Investor Relations. Please go ahead.
Hello, everyone, and welcome to our event. Before we begin, I will go over a few housekeeping reminders. There will be a moderated question and answer session following prepared remarks. To submit the written question, please type it into the question and answer panel on the webcast. The webcast replay of this event will be available tomorrow after 12:30 p.m. Eastern in the Investors section under Events and Presentations on our website, www.alector.com. I'd like to note that during this event, we'll be making a number of forward-looking statements, and you can find our disclosure here. Turning now to the agenda, we'll begin with an overview from Dr. Sarah Kenkare-Mitra, our President and Head of Research and Development. She'll share Alector's perspective on our multi-stage pipeline and our approach to driving value in treating neurodegeneration. Sarah will then provide a review of our progranulin elevating franchise in PGRN and Alzheimer's disease, highlighting latozinemab and nevisnobart, formerly AL101, which we are developing in collaboration with GSK. From there, our Chief Executive Officer, Dr. Arnon Rosenthal, will discuss advancements in Alector brain carrier and introduce our lead candidates for Alzheimer's and Parkinson's. With that, I'll now turn it over to Dr. Sarah Kenkare-Mitra. Sarah?
Thank you, Katie. Alector is dedicated to developing first and best-in-class disease-modifying therapies for neurodegenerative diseases with urgent unmet needs. We're building an integrated biotech that brings together expertise in genetics, immunology, and neuroscience with deep capabilities in discovery, development, and manufacturing. Our therapies are designed to address the root causes of disease. Our 3R strategy aims to remove misfolded proteins, replace deficient proteins, and restore dysfunctional immune cells and neurons. This strategy is powered by advanced technologies, including our proprietary Alector brain carrier, or ABC, which improves therapeutic delivery across the blood-brain barrier. With our clinical late-stage progranulin elevating antibodies in partnership with GSK, a growing pipeline of ABC-enabled programs, strong cash resources, and an experienced leadership team, we are positioned to drive both near and long-term value. Our progranulin elevating programs form the foundation of our late-stage portfolio. Latozinemab in development for PGRN has received breakthrough therapy, fast track, and orphan drug designations, and top-line phase 3 data are expected by mid-Q4 of this year. Alongside it, nevisnobart in Alzheimer's disease is fully enrolled in a phase 2 trial. In parallel, we have selected lead candidates for our ABC-enabled anti-amyloid beta antibody program for Alzheimer's disease and our ABC-enabled GCase enzyme replacement therapy for Parkinson's disease. We are also progressing ABC-enabled siRNA programs for tau, alpha-synuclein, and NLRP3 for multiple neurodegenerative diseases. This portfolio provides an important near-term milestone within our late-stage programs. It also includes the potential for the initiation of first-in-human trials from our ABC-enabled pipeline in 2026 and 2027. Let me now turn to our GSK-partnered progranulin elevating franchise, which targets frontotemporal dementia caused by granulin mutations and Alzheimer's disease. Frontotemporal dementia. While Alzheimer's disease is the most common form of dementia, you may be less familiar with frontotemporal dementia due to a granulin mutation or FTD-GRN. FTD-GRN is an aggressive, early-onset dementia. Patients often present with compulsive behavior, lack of restraint, apathy, anxiety, or aphasia. Tragically, life expectancy is typically less than 10 years, and there are no approved treatments to slow or cure the disease. Heterozygous loss-of-function mutations in the granulin gene reduce the levels of a protein called progranulin by 50%, directly causing the disease. In partnership with GSK, we are developing latozinemab, which is designed to elevate progranulin levels back to physiologic levels. FTD is rarer than Alzheimer's disease, but it is the most common cause of dementia in individuals under the age of 60, and most cases occur between the ages of 45 and 64. In the U.S., the prevalence is estimated to be approximately 50 to 60,000 people, and in Europe, the number is closer to 110,000. FTD-GRN accounts for approximately 5 to 10% of all FTD cases and represents about 8 to 17,000 cases in the US and EU alone. Importantly, the overall economic burden per patient for FTD is nearly twice that of Alzheimer's disease, underscoring the urgency of developing effective therapies. FTD-GRN is frequently misdiagnosed as Alzheimer's disease, Parkinson's disease, Lewy body dementia, vascular dementia, or unspecified dementia, as you can see in the bottom half of the table. This underscores the importance of genetic testing to ensure patients are properly identified and can access future therapies. As we have seen in other therapeutic areas, once disease-specific treatments become available, the uptake of genetic testing typically increases. That means the landscape for FTD-GRN diagnosis could shift meaningfully as potential therapies move closer to approval. Both latozinemab, being developed for the treatment of FTD-GRN, and neviznibart, or AL101, being developed for the treatment of Alzheimer's disease, are designed to increase progranulin levels by blocking sortilin, a receptor that binds progranulin and directs it to the lysosome for degradation. Progranulin, encoded by the GRN gene, is a secreted glycoprotein, which is primarily expressed in neurons and microglia within the central nervous system and has several activities, including being an immune and neurotropic factor. Human and mouse data shown on the left side of the slide demonstrate that higher sortilin levels mean lower progranulin. By inhibiting sortilin, our antibodies increase extracellular progranulin levels. The rationale for progranulin-elevating drugs in FTD is clear. On the left, we show the 50% reduction in plasma and CSF progranulin in granulin mutation carriers and patients compared to healthy controls. This deficiency triggers a neurodegenerative cascade, neuronal cell death, and microglial dysfunction, leading to TDP-43 accumulation, lysosomal impairment, complement activation, and inflammation. These processes result in destruction of brain structures, ultimately driving the cognitive and behavioral deficits seen in FTD. Latozinemab is designed to elevate progranulin levels in the brain, addressing the underlying deficiency that contributes to neuronal loss, inflammation, and cognitive and behavioral deficits in FTD-GRN. The rationale for progranulin-elevating drugs in Alzheimer's disease is also compelling. Human genetics show that loss-of-function mutations in progranulin increase Alzheimer's disease risk, while preclinical studies demonstrate that elevating progranulin can be protective. On the left, human genetic data highlights granulin as a risk gene for Alzheimer's disease. In the center, immunohistochemistry shows progranulin embedded within amyloid beta plaques in Alzheimer's disease brain tissue, emphasizing its association with disease pathology. And on the right, data from AD mouse models shows that increasing progranulin improves disease-relevant outcomes. These findings together provide strong genetic, pathologic, and preclinical support for evaluating a progranulin-elevating drug, nevisnobart, in Alzheimer's disease. The genetic and biologic rationale for blocking sortilin to elevate progranulin in FTD-GRN is grounded in multiple lines of evidence. Loss-of-function mutations in SORT1 lead to chronically elevated progranulin in humans and mice with minimal or no discernible adverse effects, providing a strong genetic foundation for this approach. Biologically, progranulin that does not bind sortilin can still enter lysosomes through alternative receptors, where it remains partially active and supports neuronal survival. You can see this in the images on the upper left. Even in SORT1 deficient cells, shown in the bottom panel, progranulin continues to traffic to lysosomes, similar to wild-type cells in the top panel. The schematic next to it illustrates how multiple receptors facilitate lysosomal entry in the absence of sortilin. Importantly, progranulin that is mutated to bypass sortilin appears more potent than wild-type progranulin in rescuing microglial pathology, reducing NFL, and correcting lipid abnormalities in mice. Experimental data further support this strategy. The graphs on the bottom left show results with nevisnobart, our antibody that blocks sortilin to elevate progranulin. In this first panel, nevisnobart nearly eliminates functional sortilin, confirming target engagement. This is accompanied by significant increases in progranulin levels in plasma and CSF as shown in the middle and right panels. And these findings demonstrate that blocking sortilin effectively elevates progranulin and restores key functions of progranulin. Together, these genetic, biological, and experimental data establish a compelling case for targeting sortilin to increase progranulin and address neurodegenerative disease mechanisms. It's important to note that latozinemab and nevisnobart target distinct regions or binding epitopes on the SORT1 protein. The PK/PD profile distinguishes nevisnobart from latozinemab. While latozinemab is being developed to be a treatment for FTD-GRN, nevisnobart's properties could make it suitable to address a broader spectrum of neurodegenerative diseases, including Alzheimer's and Parkinson's disease. Both latozinemab and nevisnobart have demonstrated a 2 to 3-fold increase in progranulin levels and have been generally well tolerated in clinical trials to date. Turning now to clinical data in our progranulin elevating antibodies. I will begin with our INFRONT-2 phase 2 study of latozinemab, which was designed to gather data on safety, PK, PD, clinical outcomes, and biomarkers. In the trial, 12 symptomatic FTD-GRN patients were treated with latozinemab at 60 mg/kg every 4 weeks for 49 weeks. To determine whether there was treatment-related slowing of disease progression, we used historical data from the GENFI2 and ALL-FTD non-interventional registry databases to generate a matched control cohort that would allow us to make comparisons to FTD-GRN participants in our open-label interventional cohort. In INFRONT-2, we evaluated 3 areas: target engagement through progranulin levels in plasma and CSF, biomarkers of disease activity, including lysosomal function, inflammation, brain health, and atrophy, as well as clinical progression measured with CDR plus NAC STLD, a tool designed for FTD. We'll now review some of the key findings. In INFRONT-2 study, latozinemab increased progranulin in plasma and CSF 2 to 3-fold, restoring levels to those seen in healthy controls and sustaining them over 49 weeks. Also, Plasma and CSF concentrations were strongly correlated, supporting the use of plasma progranulin as an important biomarker for use in clinical trials. Here we show the in-front-2 data for GFAP, or glial fibrillary acidic protein, a marker of astrogliosis that is elevated in symptomatic FTD-GRN and correlates with disease severity. These figures show that GFAP levels are elevated in symptomatic FTD-GRN patients at baseline, and that at treatment, GFAP levels decline over the course of the study towards the range seen in asymptomatic GRN mutation carriers. We also looked at disease progression using the CDR plus NAC FTLD, a measure of clinical progression in FTD agreed upon by the FDA and EMA. In matched controls from the GenFi2 registry, patients declined by 6.4 points over 12 months. By contrast, participants treated with latozinemab declined by 3.1 points over the same period, which represents an estimated 48% slowing of clinical progression compared to matched historical controls. In INFRONT-3, our pivotal phase 3 trial, we are measuring the same clinical measures and core biomarkers that we assessed in our INFRONT-2 phase 2 trial. INFRONT-3 is a 96-week randomized double-blind placebo-controlled global trial evaluating latozinemab in 103 symptomatic and 16 at-risk individuals with confirmed GRN mutations. Participants receive 60 mg per kg or placebo via intravenous infusion every 4 weeks. The primary analysis will be conducted in symptomatic participants, and we plan to include at-risk participants in a sensitivity analysis. The clinical co-primary endpoint is the CDR plus NAC FTLD sum of boxes, and in the US, the biomarker co-primary endpoint is plasma progranulin. Additionally, we are measuring key secondary outcomes assessments and collecting fluid and imaging biomarkers, including plasma NfL, GFAP, and volumetric MRI. We believe this positions us to deliver a clear and well-aligned data package later this year. Our phase 2 study of nevisnobart in early Alzheimer's disease is ongoing. In partnership with GSK, we completed enrollment of that study in April. An independent interim analysis is expected in the first half of 2026. Progress AD is a global, randomized, double-blind, placebo-controlled phase 2 clinical trial enrolling patients with early Alzheimer's disease. The study is designed to assess the safety and efficacy of 2 dose levels of nevisnobart compared to placebo. Participants are randomized to receive nevisnobart or placebo intravenously every 4 weeks for the duration of the 76-week trial. The primary endpoint of the study is disease progression as measured by the Clinical Dementia Rating Sum of Boxes, or CDR Sum of Boxes. We are also measuring key secondary endpoints and biomarkers, including amyloid PET, tau PET, and biomarkers in CSF and plasma. Our progranulin programs, latozinemab and nevisnobart, are being developed in partnership with GSK. This partnership included $700 million in upfront payments and includes a $1.5 billion in potential development and commercial milestones, a 50-50 U.S. profit share, and tiered double-digit royalties ex-U.S. Potential milestone payments include $160 million for the first commercial sale in the U.S. and $90 million for first commercial sale in at least 2 of the EU countries. With that, I'll turn the call over to Arnon to discuss our preclinical programs enabled by the Alector brain carrier.
Thank you, Sarah, and welcome everyone. I will now guide you through a tour to our imminent future, which is propelled by Alector brain carrier. Alector brain carrier is using the transferrin receptor on the blood-brain barrier endothelial cells to deposit and transport cargo to the brain. There are 3 features of Alector brain carrier that are worth mentioning. First, is that our brain carrier module is completely independent. It can be placed in multiple places on the target. On the left here, you see placing the brain carrier on the constant region of the antibody with a linker. The linker can be of any size or no linker at all. In the middle figure, you see us placing the brain carrier model as part of the FAB configuration. And on the right, on the bottom, you see that we can place the brain carrier as part of a single ARAM antibody or as part of an FAB. Our brain carrier can be bivalent, as you see on the left bottom, or monovalent. So this flexibility really enables us to tailor the drug to the different drug modalities, be it antibody, enzymes, or nucleic acids. The configuration of the drug really dictates the drug half-life, immunogenicity, and brain penetration. So this is an important tool to optimize blood-brain barrier-propelled drugs. The next feature of Alector brain carrier is the large variability or the large range of affinities that we can deploy. As you see on the left table here, we have almost 1,000-fold range of affinities, from 5 nanomolar to almost 5,000 nanomolars. And again, the affinity dictates hematologic adverse effects, drug half-life, and brain penetrance. And this large range of affinity enables us to tailor the affinity to each drug modality, and specifically within each drug modality to each drug, and gives us a real unique tool to optimize ABC-propelled drugs. The third unique feature of our platform is the epitope that they are using to engage the transferrin receptor. Whereas many blood-brain barrier technologies are using an epitope which we consider to be exposed on the transferrin receptor, we have identified a different epitope that is depicted on the left side of the structure of the transferrin receptor. And this epitope is between two lobes, and the uniqueness of this epitope is that it is reducing the ability of ABC-propelled drugs to induce transferring and antibody-dependent cell cytotoxicity. And this is apparent on the right graph. And you see in the red line, using the exposed epitope, what we call an epitope B, which many other companies are using, and this enables the antibody to facilitate ADCC that is transferring receptor dependent. In contrast, when we use our own epitope, that's depicted on the graph in green and yellow here on the right, you see a significantly lower ADCC. And this really provides a unique safety feature for our drugs. We think that reduced ADCC would translate to reduced hematologic adverse effects and increased safety of our ABC module. With these ABC features, we are developing, as Sarah mentioned to you, 3 types of drug modalities. We are developing antibody drugs that are propelled by ABC, enzymes that are propelled by our ABC, as well as multiple siRNAs that are propelled by ABC. I will start by describing our antibody against the beta amyloid that's an ABC enabled. Every aspect of our anti-A-beta antibody, which we designate as AL037, was engineered to optimize efficacy and tolerable pharmacokinetics and safety. So the anti-A-beta binding epitope was engineered to recognize the pyroglutamate version of the A-beta peptide, as was shown by Lilly. Targeting the pyroglutamate A-beta was the most effective in reducing A-beta plaque rapidly, and there is no A-beta pyroglu in the circulation in the serum, so this is minimizing sink effects and retention of the antibody in the periphery. we undertook or chosen to retain a fully functional effector function. We think that recruiting myeloid cells through the effector functions is critical to fully remove beta amyloid plaques, and that the effector function cannot be substituted by the transferrin receptor or by a crippled effector function. So we are targeting to maximize removal of A beta plaques with a full effector function. We then added to this drug our optimized ABC technology, which is designated to maximal brain penetrations and minimal hematologic adverse effects, as well as maximize pharmacokinetics in the serum. And I will now show you some data with this drug. The first thing that we did is to confirm that L037 can really transcytose through brain endothelial cells. And on the left side, you see a cell culture assay with brain endothelial cells, where you can put the drug on the top, on the apical side, the blood side of the culture, and then you can measure how much of the antibody can move to the basolateral side, the brain side. You can see in the middle panel here that naked L037, this is an antibody that does not have the ABC module, is not able to cross through the endothelial cells. In contrast, L037, which does have the ABC module, very readily being transduced through the endothelial cells, and this is quantified on the graph on the right. A very significant part of the antibody reaches the basolateral of this culture, of this transferred culture, suggesting that it will be able to translate those to the brain in vivo. The next thing that we confirmed was the ability of L037 to phagocytose A beta peptides. And on the left side here, you see images of human microglia phagocytosing fluorescent A beta and internalizing it. And again, you see that L037 is very effective in phagocytosing A beta. And on the right side, there is quantification of this phagocytosis event. You see that naked L037 in orange here can phagocytose A beta plaques, but the blue, which represent AL037 with the ABC module, can phagocytose A beta plaques even better. And this suggests that the ABC module can contribute to phagocytosis, but it's still incremental, and you still require a full effector function, including the A beta gamma receptor for full phagocytosis. We next moved in vivo. We first tested L037 in mouse models of Alzheimer's disease. This is the 5XFAD mice. You see on the left picture, this is the light sheet microscopy. You see that the naked antibody primarily gets stuck in the ventricle in large blood vessels and is not distributed well into the brain. In contrast, the three right graphs shows antibodies that are enabled by our ABC. And in all cases, you see that there is no longer stickiness into ventricles and blood vessels. There is homogeneous distribution in the brain. On the two right panels, you see that our antibodies can detect A beta plaques very well and really bound to them. And the most right pictures show a three-dimensional reconstitution. You see antibodies recognize practically all the plaques in this brain, or many of the plaques, and can sort of recognize and bound to them. L307 not only bound to plaques, it also stimulates microglia-dependent removal of plaques, and this is seen on the graphs on the right. You see that even after 3 to 4 injection of the antibody, in a very short time frame, you see significant reduction in the level of A beta 42 in the brain. So antibody at fairly low doses is able to penetrate the brain, distribute homogeneously, and remove A-beta-42 in a very short duration. With the mouse data at hand, we advanced to non-human primate studies. The first thing that we looked at was the half-life of AL037, and you see in 2 doses, 30 mg per kg and 3 mg per kg, we see that the half-life of AL037, which is linked to our ABC, is about 106 hours. And this is significantly better than reported brain shuttle-linked antibodies, and it's actually not far off from a naked antibody like a normal antibody. So we do show a very decent half-life of the antibodies in non-human primates. The next thing that we looked at was hematologic side effects. As you know, there is 10 times more transferrin receptor expressed on the reticulocytes compared to endothelial cells. So all brain shuttle technologies that are using transferrin receptors have an inherent risk of hematologic side effects. So we measured that. As you see on the left graph, we do see transient reduction in reticulocytes, but the reticulocytes recover very quickly within a day. And even after 2 injections at day 1 and day 8, we don't see a meaningful reduction in red blood cells in the middle graph or in hemoglobin in the right panel. So we see a very tolerable safety profile for AL037. We next looked at brain penetration in the non-human primates. And to our delight, we saw very potent brain penetration. As you see on the left panel in the frontal cortex, even at 3 mg per kg, which would be around the range of the clinical doses, we see 18-fold elevation in brain penetration, and we see it in every brain region that we tested. And here we show just the frontal cortex on the right, on the left, and hippocampus on the left, but there is a homogeneous distribution of AL037 in the non-human primate brain. If you calculate the molarity of brain level, you see that our AL037, even at 3 mg per kg, can reach a 3.8 nanomolar in the brain. And this is, according to our calculations, at least 5 times higher than what was reported for other brain shuttle antibodies that are currently in the clinic. So we see a very potent high concentration in the brain that suggests that this antibody would be very effective at removing A-beta plaques in humans. Given the significance of Alzheimer's disease as an unmet medical need, we are developing a second anti-A-beta antibody, which we designate as AL137. This antibody is deploying a different ABC modality with higher affinity, and we see that in this case, we can achieve 32-fold increase in brain level. And this 32-fold increase can translate to 8.4 nanomolar even at 3 mg per kg in the brain. And this is over 12 times higher concentration that what, according to our calculations, was reported for clinical brain shuttle anti-A-beta antibodies. So we have, again, an even more potent anti-A-beta antibody, which have still very, very good pharmacokinetics and tolerable safety features. To summarize what I've shown you on our anti-A-beta programs, we have 2 anti-A-beta antibodies, AL037 and AL137, that target the pyroglue anti-A-beta peptide, which in our view, and as was demonstrated by Lily, is the most potent epitope for removal of A-beta plaque. We retain a fully active FC region to enable full function of recruitment of myeloid cells to remove A beta, and we have an optimized ABC module that can lead to high brain penetration, but still good hematologic safety and good pharmacokinetics. So we think that we have two uniquely potent and safe antibodies that could really be best in class in anti-A beta therapeutics, and we are targeting first in human in 2026. The next program that I will describe is our brain carrier-enabled GCase enzyme replacement therapy for Parkinson's disease and eventually Lewy body dementia. As you know, GCase or GBA is a lysosomal enzyme that removes toxic substrates, toxic lipids like glucoceramide and glucose sphingosine from cells, and if you don't have functional GCase, this toxic lipids accumulate and cause diseases. There are up to 10 to 15 million people that have Parkinson's disease that carry the GCase mutations. This translates to up to 1.5 GBA mutation carriers with Parkinson's. There are up to 2.4 million GBA mutation carriers that suffer from Lewy body dementia, and there are over 100,000 Gaucher disease patients with the GBA mutation. There is, as you know, currently there is enzyme replacement therapy for Gaucher disease, which display peripheral symptoms, but there is no enzyme replacement therapy for Parkinson's disease or Lewy body dementia because current enzyme replacement therapy cannot enter the brain, and even if it enters the brain, the way it enters cells does not allow it to enter as nerve cells or myeloid cells in the brain. Even though we are starting with genetic mutation carrier, there is really good evidence that GCase enzyme replacement therapy could be also beneficial for the sporadic form of Parkinson's disease and likely Lewy body dementia. The reason is that if you look at all-comer Parkinson's patients, if you measure the level of GCase activity versus the rate of disease progression, you see in the yellow line on the left graph that people that have high level of GCase activity show low progression rate, whereas people that show low level of GCase activity in the dark line display very high progression rate. And consistent with the level of enzymes you see on the right graph here, in blue, people that have low level of the toxic lipids show slow progression rate, whereas people with Parkinson's disease, regardless of whether they carry the genetic mutation or not, people that have high level of the toxic lipids display very rapid progression rate. So this really suggests to us that even in sporadic form of Parkinson's disease, GCase, brain-penetrant GCase enzyme replacement therapy could be beneficial to slow disease progression. So as we did with AL037, we did for AL050, we engineered every component of the drug. We first engineered the enzyme itself. The wild-type GCase enzyme has very short half-life. It is very unstable, and it's hard to manufacture. So we engineered GCase that is almost 30-fold more stable than the wild-type. You can see it on the table on the left bottom. You see that wild-type enzyme has a half-life at 37 degrees of 6 hours versus 7 days of our enzyme. Likewise, we engineered the enzyme to have almost 50-fold higher activity. You see this is a logarithmic scale. You see the activity of the wild-type enzyme versus AL050. So we designed a very potent and stable engineered GCase as the first component of the drug. We then linked it to an optimal ABC that has a higher affinity that enables fast removal, fast transport to the brain, and as I'll show you, also transport to cells and to the lysosomes in brain cells. And finally, because we don't need to recruit immune cells, we silence the effector function of this drug to minimize or eliminate hematologic-related adverse effects. So we took this drug initially to testing in cell culture, and the first thing that we did was to confirm that AL050 can enter lysosomes in cells. As you may know, the peripheral enzyme replacement therapy is using macrophages Mannose receptor to enter cells. So current enzyme replacement therapy for GCase, the protein can only enter macrophages in the periphery. It cannot enter nerve cells, and it cannot enter microglia cells or other cell types in the brain. So first we wanted to see whether we can substitute the Mannose receptors with the transferrin receptor, and we saw that this is indeed the case. You see here that our GCase that is propelled by ABC can enter the lysosomes, and you see overlay of staining, fluorescent staining with lysosomal markers like LAMP1. So on the left side, we show that our GCase propelled by our ABC can enter cellular lysosomes, and these are neuronal cells that the regular enzyme replacement therapy for GCase will not be able to do. On the right side, we also looked at activity of the GCase in this lysosome, and we see that there is very potent GCase activity in these GCase deficient neuronal cells. And the level of activity is transferrin affinity dependent. So the higher affinity, the ABC model, the higher the activity. And you see that with affinity that we are using in the for the clinical program, we are easily exceeding the wild type level of GCase activity, which is marked in the gray horizontal bar on the right graph. So based on the cell culture activity, we found out that we have a very potent and active enzyme, and we have a drug that can enter lysosomes in neuronal cells and can retain activity in neuronal cells. With this information, we took our drug to non-human primates. We first test the half-life of our drug in the non-human primate plasma. And as you see on the left graph, AL050 display a half-life of 5 hours. And this is compared to current enzyme replacement therapy that's sort of illustrated on the right side of this slide. Current enzyme replacement therapy displays a half-life of less than 30 minutes. So in this experimental paradigm at least, AL050 displays tenfold higher, longer half-life in the plasma compared to current enzyme replacement therapy. We then looked at the enzymatic activity in the non-human primate plasma, and we see on the left side here that AL050 delivered at 10 mg/kg peripherally display half-life of activity of 6.6 hours. And again, this is almost 40-fold longer than the enzymatic activity that was reported for current GCase enzyme replacement therapy, where the serum half-life of enzymatic activity is about 10 minutes at the maximum, even in humans. So at least with these experiments, we show that both in vitro, biochemically, in cell culture, and in non-human primates, we have a stable and an active drug that retain enzymatic activity extracellularly and sort of this will allow time for the drug to enter the brain and possibly retain activity in the brain. And these are the things that we looked at next. And before we did that, we sort of looked at the safety of AL050. Again, all brain shuttles that use transferrin as the Trojan horse have a risk of hematologic side effects. So we wanted to make sure that this is not the case with 050. You see on the left graph here, AL050 does not lead to any meaningful reduction in reticulocyte count after 2 injections. Likewise, there is no effect on red blood cell count and no effect on hemoglobin in this experiment, suggesting that AL050 will be safe with regard to hematologic adverse effects. We next looked at what happens in the brain. So the first thing that we looked at was whether our ABC-propelled AL050 can actually enter the brain, basically cross the blood-brain barrier and enter tissues in the non-human primate brain. And we see that this is indeed the case. You see that in every brain region that we looked at, the frontal cortex, hippocampus, as well as brain regions that are relevant for Parkinson's disease and Lewy body dementia, the substantia nigra, where the dopaminergic cell body resides, and the putamen, where the dopaminergic nerve endings are. And we show very good enzymatic and very good level of AL050. That's sort of somewhere between 6 and 20-fold elevation of enzyme. We then looked at enzymatic activity, and again, for enzymatic activity, our AL050 has to enter the brain, has to enter neurons and support cells in the brain, has to enter lysosomes in these cells, and then has to retain activity. So this is a very significant demand for a drug, and we were not sure that this would happen. But to our sort of delight, it did happen. And you see that in all brain tissue that we looked at, again, including the substantia nigra and the putamen, which are relevant brain regions for Parkinson's disease and Lewy body dementia, we see at least 2-fold elevation in GCase activity. Parkinson's patients and Lewy body dementia have modest reduction in their GCase activity, somewhere between 15 and 50% reduction. So doubling the level of GCase activity would be more than sufficient to fully repair enzyme deficiencies in these diseases. And moreover, we think that there is some negative regulation feedback on lysosomal enzymes. So in healthy brain, like in the non-human primate brain, high level of GCase enzymatic activity could lead to reduction in the level enzymatic activity of the endogenous GCase. So we think that in patients, this level could be even higher. Although, again, as I mentioned, 2-fold elevation is significantly more than what is needed to fully restore GCase deficiency in Parkinson's patients and Lewy body dementia patients. We then wanted to see whether our drug can actually correct disease pathology, and for this we went back to mice. These are mice that carry one of the Parkinson's GBA mutations, the D40-409-2V changes, and this mutation reduces GCase activity by 85%. So on the left bar graph, you see the level of GCase in wild-type mice. This is in the gray. You see on the right side of the left panel, you see that the mutation curves, the mouse mutation curves that were treated with PBS do not show hardly any GCase enzymatic activity. However, once we inject AL050 serodat, we see almost complete restoration of GCase activity. And this is just 24 hours after one injection, you see almost complete restoration of enzymatic activity. We went further and looked whether the increase in enzymatic activity is also associated with reduction in toxic substrates, and this was done initially in the periphery, in liver. I'll show you data in the brain in the next slide. So again, you see on the right panel, the right graph, wild-type animals do not have toxic substrates because the endogenous GCase gets rid of them. However, GBA mutation carriers have a very high level of toxic substrate, as you see from the blue bar graph here. But even sort of 2 injections of AL050 surrogate reduce these toxic substrates by almost 90%, suggesting that at least in the periphery here, our drug is very active, can enter cells, and can restore enzymatic activity and can remove the toxic lipids that cause the diseases. We then wanted to see if the same thing can happen in the brain of this GBA mouse mutant. And what we found out is that this is indeed the case. You see on the left panel of bar graphs, this is still wild-type mice that carry the human transferrin receptor, but they have a wild-type level or normal level of GCase. You see with PBS, this is in gray, this is the normal level of GCase. But if you inject AL050 surrogate, you almost double the level of GCase activity in the mouse brain. We then went to GBA mouse mutants. Again, in the middle graph, you see that wild-type mice injected with PBS does not show any toxic substrates because the endogenous GCase take care of these. In contrast, GBA mutant mice show high level of toxic substrates, as you see from the blue bar, and a single or 2 injections of AL050 surrogate reduce the toxic substrate by 80%. And again, the enzyme will continue to work, so 2 injections is a very short and acute timeline. We think that over time the reduction will be even more profound. Although 80% reduction in the toxic substrate, we think could be significantly therapeutic. We wanted to look at the durability of the effect on the right panel here, and we see that even after a single injection, the reduction in the toxic substrate can last for over 14 days, suggesting that once our enzyme gets into the lysosome, it retains activity for multiple weeks, way beyond the residence in the plasma. So just to summarize what I've told you, we have engineered AL050 to have multiple beneficial drug features. It has an engineered GCase that is 30-fold more stable and 50-fold more active than the wild-type GCase, that shows long half-life and long enzymatic activity in the non-human primate serum. We show that AL050 can enter the non-human primate brain, enter nerve cells and support cells in the brain, enter the lysosomes, and retain enzymatic activity. So we think that we have a pretty potent drug that could be beneficial for, again, Parkinson patients that carry the GBA mutations, Lewy body dementia patients that carry the GCase mutations, and eventually sporadic forms of these diseases. And we are targeting first in human in 2027. I will just now describe our progress with Alector brain carrier-enabled siRNA programs. As you know, siRNA is becoming a very successful drug modality. I think that there are over 20 approved siRNA or ASO drugs. Now, most of them are for peripheral indications. There are 2 nucleic acid drugs that are approved for central indications, for ALS and for spinal muscular atrophy. The issue for nucleic acid or siRNA for brain disorders is that you have to deliver it by IT or ICV injection. This is a surgical process that is not safe. It's not highly scalable for large diseases like Alzheimer's disease. And also with IT or ICV delivery, the nucleic acid is not distributed equally throughout the brain. So some brain regions which are close to the injection sites receive more nucleic acid drugs, whereas regions which are further away from the injection sites receive, or deep in the brain, receive maybe insufficient drug. So we are undertaking to change that by enable peripheral delivery of siRNA using our ABC technology. So we screened very extensively, as you see on the second from the left column. We screened very large range of transferring affinities with different types of linkers, with different types of drug modalities, to identify a good ABC carrier for siRNA. And this was really enabled by the versatility of our technology where we can put the ABC module in everywhere practically on the drug. We can use linkers or not use linkers. We can use cleavable linkers or non-cleavable linkers. We can use different affinity and we can use different valency. And this has really enabled us to optimize the siRNA delivery. As you see on the third column from the left, we are able to achieve over 40-fold increase in siRNA delivery in the brain following peripheral injection. And the first thing that we did was to look at superoxide dismutase as a proof of concept strategy. And again, with siRNA to superoxide dismutase, we see with different ABC modules, we see 30 to 40-fold elevation of siRNA in the brain. And we also see, as depicted in the right column, very good long plasma half-life. We then wanted to see whether our ABC propelled siRNA enters the brain, enters cells in the brain, and enter the cytoplasm of the cells and is really able to suppress, to downregulate siRNA. Again, these are very demanding requests from a drug. We inject the drug peripherally. It has to go through the endothelial cells, like to 2 membranes of the endothelial cells, to go through membranes on nerve cells and support cells, and to enter to the right subcellular localization to access the messenger RNA for, in this case, SOD, and to downregulate it. And to our delight, we saw that our drug is able to do that. If you see every brain region that we looked at, the thalamus, cortex, hippocampus, brainstem, cerebellum, spinal cord, and striatum, in all cases, we see reduction in the SOD mRNA by up to 80%, and this is depicted by the purple and blue graphs that represent our peripheral injected siRNA with 2 different ABC modules. The purple graph represents ICV injected naked siRNA, and you see that in all cases, peripherally injected siRNA with ABC is at least as good as ICV injected siRNA, and in most cases it's significantly better. If you see in the cortex, in the spinal cord, in the brainstem, you see that our peripherally delivered siRNA is better than siRNA injected ICV. So this really tells us that we could peripherally deliver siRNA, achieve homogeneous distribution in the brain, convert surgical procedure of ICV or IT into either easily delivered infusion centers or ultimately even at home delivery with IV or even subcutaneous delivery. So we think that there is significant potential for this technology to increase ease of use, safety, and also efficacy. With this technology, we are now developing 3 programs. We are developing tau siRNA with ABC for Alzheimer's disease and frontotemporal dementia that is caused by tau pathology. We are developing alpha-synuclein siRNA for Parkinson's disease and Lewy body dementia. And we are developing NLRP siRNA for multiple neurodegenerative diseases. As you know, NLRP is an inflammatory mediator that is thought to be involved in practically every neurodegenerative disease, from Alzheimer's disease, Parkinson's disease, ALS, Huntington's disease. And so far, small molecules for NLRP were not as effective in the clinic, partially because of off-target activities and partially because of incomplete blockade. So we think that all of these 3 programs have a very profound potential in very large diseases. Just to summarize what I've told you today and what you heard from Sarah, we have a good mix of late stage and late preclinical programs at Alector. Our late stage programs have a program in phase 3 that is a pivotal phase 3 where we received breakthrough therapy, orphan designation, and fast track designation. And we will have data in the middle of Q4 2025. And because of the designations that the drug gets, we think that we can proceed. If the drug justifies that, we can proceed to BLA rapidly and to commercializations. And we have commercial rights in the US, so we will be leading commercialization of this drug, US, whereas our partner will lead commercialization ex-US. Our second drug, as Sarah told you, is in early Alzheimer's disease. It's sort of completed recruitment. It's a 76 in April. It is a 76-week-long trial. So if you calculate, you see that trial completion is in 2026, and we expect to have data shortly after. In addition to our promising late-stage programs, we have, as I described to you, multiple late preclinical programs that involve antibodies that are propelled by our ABC. I described the anti-beta antibody, but we also are advancing anti-tau antibody. We have enzymes that are propelled by ABC for Parkinson's disease, Lewy body dementia, and eventually Gaucher disease with neurological pathology. And we have an emerging platform of siRNA that are propelled by our ABC. These include currently siRNA for tau, for alpha-synuclein, for NLRP, but if successful, there are many more targets that would benefit from our ABC module. Again, as Sarah described to you, we are completely focused on neurodegeneration. We are going after the core of the disease by developing tools to replace damaged or mutated proteins with enzyme replacement therapy, removing misfolded proteins with antibodies and siRNA, and restoring damaged neurons and support cells by antibodies that stimulate signaling in these cell types. We have still over $300 million in the bank that will enable us to complete the two late-stage programs and to take at least two of our preclinical programs to first in human. We are targeting, again, to have multiple catalysts in both late and early stage programs in the next 1 to 2 years. So thank you everyone for listening, and we will now open the webinar for questions.
Thank you so much. And as a reminder, to ask a question, simply press *11 on your telephone. To remove yourself, press *11 again. One moment for our first question. It comes from the line of Pete Stavropoulos with Cantor Fitzgerald. Please proceed.
Hello, and thank you for hosting this event. Very informative and great to see all this activity across the pipeline. First question has to do with Infront 3, as you have the phase 3 reading out, and it's top of mind. Can you just discuss some of the conversations and data that you provided to the FDA that helped enable progranulin as a co-primary endpoint, and the sense that you received that they could possibly lean on it for an approval if clinical outcomes were trending in the right direction, but not stat sig?
Thanks, Pete. I think I'll have Giacomo address this question. Giacomo?
Thanks for the question. The ask by the FDA to move progranulin as co-primary endpoint came as part of the review of the statistical analysis plan that we submitted several weeks ago. We didn't submit any new data to justify this recommended change by the FDA. Previously, in 2024, we discussed the use of progranulin as confirmatory evidence in addition to a single pivotal phase 3 study, and the FDA had agreed upon. And this is a change that stemmed from this original discussion, but again, no new data were submitted by the company.
All right. Have you previously shown them the correlation between plasma, because I'm specifically the co-primary endpoint is plasma progranulin versus CSF?
We hadn't shown the correlation, but we had presented as part of the breakthrough designation package the results on elevation of progranulin in plasma and the CSF that were comparable in magnitude.
Right. And one question, please, on the amyloid beta ABC program, first in human trial in 2026. I know it's early, but not in the clinic yet. But curious to hear how you're thinking about clinical development, sort of leveraging the data generated by approved amyloid beta antibodies and, those in development that can cross the blood-brain barrier easily with technology. What would a proof of concept study sort of look like, and what would you like to see to move forward into late stage programs? Would it be just imaging data or clinical data? And when it comes to safety, there were very low levels of ARIA for trontinemab. Does that molecule, do you know if that molecule has an active FC region? And what are your expectations for your amyloid beta ABC? Will that translate to low ARIA rates and safety translate?
Maybe Giacomo can address the first part of the question, particularly in terms of what we believe a proof of concept would be for our anti-amyloid beta ABC.
Sure. The most recent data presented by other companies have shown that using biomarkers such as amyloid PET, it's possible to derive anti-amyloid treatments relatively fast and relatively early in the development program. So the current thinking is to use a similar approach and use extensively biomarkers, amyloid PET, as well as the p-tau species in plasma to have information about the target dose and have an early read on pharmacodynamic effects that are important for therapeutic benefit. Regarding the other question was about what we are expecting to see, and we will design studies to provide evidence of very meaningful and fast amyloid clearance, but using both PET biomarkers as well as fluid biomarkers.
Yeah, so just to add to this, there's very clear path for anti-A-beta drugs now. As Giacomo said, like, we want to see profound reduction of A-beta plaques within 3 to 6 months. We want to see minimal ARIA, and Roche is also using a full effector function, so we don't think that will impact the ARIA. I think the ARIA is impacted by the way the ABC or the blood-brain shuttle enters the brain. So we expect minimal ARIA, and we also are hoping to see a low level of infusion reaction, something that was not reported with sort of competing antibodies. So we think that there is very clear sort of proof of concept for our drug that can happen with a fairly small clinical trial fairly quickly.
All right. Thank you very much for answering my questions, and I'll hop back in the queue.
Thank you. Our next question comes from the line of Myles Minter with William Blair. Please proceed.
Hey, thanks everyone for taking the questions. The first one on INFRONT-3 again. I know you've only enrolled, I think it's 16 asymptomatic patients in INFRONT-3. It's not part of the primary analysis, but is there a path forward for those asymptomatic patients to get them on label if you do see positive data from INFRONT-3 in the symptomatic cohort? That's the first one. The second, I'm curious as to the comment that the progranulin efficacy on lysosomal function is actually enhanced if it gets uptaken through LRP1 versus sortilin-1. If you could just expand on that, because that is a source of investor concern that you wouldn't actually get active granulin concentrations in the lysosome itself if you block SORT1. So that's the second one. And the third one is just coming back to Pete's question. If you're going for fast amyloid removal with your amyloid antibody with the ABC carrier technology, is it safe to say that you'd want to see greater than 91% amyloid clearance at 6 months that we did see from the trontinemab data out of AAIC with the high dose? Thanks very much.
Thanks, Miles. Maybe I'll address the first question and then pass it to Arnon to answer your progranulin biology question, and to Giacomo on the anti-amyloid beta. In terms of INFRONT-3, as you asked, you're correct, Miles, that we have 16 asymptomatic carriers that we have data on. And as of now, we have not had any direct discussion with the agency in terms of label and what that data, the impact of that data will be. As I said, we will be doing a sensitivity analysis based on data from these patients. And then depending on what we see, we would have further conversations on the label with the agency. I will pass it to Giacomo to speak about to Arnon to speak about the progranulin biology question first.
Yes, Miles. So this is a recent publication by an academic lab where sort of they compared actually with sort of gene therapy progranulin that's mutated that does not bind sortilin and progranulin that can bind sortilin, and they show that the like in mice that are deficient in endogenous progranulin, and they show that the progranulin that does not bind sortilin is actually more potent in multiple aspects. It can reduces neurofilament, whereas the wild type progranulin was not able to do that. It can reverse multiple lysosomal pathologies more potently than the wild type progranulin. It is surprising, but that's what they reported, and I'm happy to send you the publication. And sort of that's a sort of second publication from this group showing that progranulin that cannot bind sortilin can restore both intracellular pathology, primarily, again, lysosomal pathology in microglia, and overall disease pathology.
Giacomo, do you want to address the
Sure.
A-beta question?
Absolutely. So, Miles, we are very pleased to see the results that Arnold presented about effects of AL037 in non-human primates, which shows very strong brain penetration. We aim with this program to show evidence of fast and very meaningful reduction of amyloid in the brain, along with a safety profile which is favorable, meaning very low frequency of ARIA, no significant concerns around anemia, and potentially lower number of infusion-related reaction, and as well as developing a compound that lends itself to subcutaneous administration as part of the development program. We don't think that we necessarily need to have a greater efficacy in removing amyloid than 91% that you cited with trontinemab. Alzheimer's disease is a very large population with very significant unmet needs, and I think these are the goals of the program, not necessarily showing superiority, which I don't think is needed at this stage.
Makes sense. Thanks for the questions.
Thank you. Our next question comes from the line of Tom Schrader with BTIG. Please proceed.
Thanks for the very informative session. Just to the last point, shouldn't a sortilin or a progranulin that can't bind sortilin be more active? It's essentially stabilized. So doesn't it make complete sense?
Yes, in our mind, it does make sense, but as sort of Miles just alluded to, there is still raging debate in the field whether how essential sortilin is for the function of progranulin. And despite the fact that there is a wealth of evidence that progranulin can enter the cells without sortilin, enter lysosomes, retain functionality, both in cell culture and in mouse models in vivo. And despite our phase 2 data, the open label data showing that all the parameters sort of go in the right direction, cognition, biomarkers, imaging. So for us, it makes sense, but yes, hopefully the actual phase 3 data will convince the doubtful.
I get it. Okay, and then with all your work in shuttles, you must, or it seems likely you've made either a shuttle direct progranulin or a shuttle sortilin receptor. Do either of them behave better in the gazillion assays you must be able to run, or do you think 01 and 101 are for you going to be good enough forever?
Yes, we are sort of developing second generation programs for everything that we do. So far, we think that 001 and 101 are looking really good. Means the issue with enzyme replacement therapy for progranulin is controlling the dosing. Like progranulin is a growth factor. We have a built-in safety feature that we cannot elevate progranulin more than 2 to 3-fold, which we think is a safe domain. Like if you do enzyme replacement therapy, you can elevate progranulin in the periphery way more than that, and that could really lead to adverse effects. So we think that there is safety issues, like one of the companies that developed enzyme replacement therapy for progranulin had to sort of show significant immune-related adverse effects and had to use immunosuppressives to deliver the drug. Our drug seems to be very well tolerated to the point that we think it will be applicable for prevention therapy if the data justify that. So far, we think that we have a really good drug, but we are always developing second generation drugs. We think that the target interesting.
And then the last one on the siRNAs, you gave a relative amount of delivery relative to systemic of 40-fold, but do you know you have enough? You kind of know how much siRNA you need outside of cells in primates or something very close to humans. Do you know you're getting enough siRNA in? And in some sense, are you out of the woods with PK for siRNAs?
Yes, in the rodent, all the experiments were done with equal molarity with the ICV and peripheral injection. So we think that we get and we see over like up to 80% suppression of the SOD mRNA. So we think we are getting enough siRNA to the brain. We are sort of in the midst of sort of non-human primate studies. We'll see how it's translatable, but so far, what we see is that we can get enough siRNA both to peripheral tissues and to the brain.
Okay, great. Thank you.
Thank you. One moment for our next question. And it comes from the line of Yaron Werber with TD Securities. Please proceed.
Thanks for the great event. This is Steven on for Yaron. A few questions here. So for the anti-amyloid beta ABC program, you mentioned first in human by 2026, and you discussed two molecules, AL037 and 137, which seems to be new. And you highlighted a few differences in the half-life. So what accounts for those differences? Are these two different molecules with different epitopes, or is this same antibody with a different ABC carrier that might affect the PK/PD? And then secondly, just to clarify, are you still deciding between 037 and 137, which look pretty similar? And how would you make the decision of what to take into the clinic?
Yes, the difference between the two antibodies is the ABC module. I mean, they have different affinity, and the affinity dictate both the sort of level of brain penetration and the pharmacokinetics. So the difference in pharmacokinetics is sort of target-dependent clearance in the periphery. So they have different pharmacokinetics. It's not clear what's the optimal peripheral pharmacokinetics for anti-A-beta antibodies, but we think that we have two very, very potent and promising anti-A-beta antibodies that are driven by two variants of our ABC. At this point, we are advancing both of them, and if there will be, we will continue to explore. If there will be data that will clearly point to one of them, we will advance one. But at this point, we think that both of these antibodies are very promising. They have different features that are worth examining, and we are committed to advance both of them at this point.
Understood. And maybe finally, you mentioned an interim data update of Vizamyl in Progress AD in the first half of next year. What can we expect to see from that in terms of patients followed or time of follow-up?
So I can respond to that. The interim analysis planned in the first half of 2026. We haven't actually divulged the exact details of the interim analysis, and we shall be doing that in the future.
Thank you very much.
Thank you so much. One moment for our next question. It comes from Paul Matteis with Stifel. Please proceed.
Hi there. This is Julian for Paul. Thanks so much for taking our question, and appreciate you hosting this very informative event. Could you just describe, have you ever shared beyond the 12-month data that you've disclosed for INFRONT-2, where you've had 12 patients compared to the 10 patients in the longitudinal FTD registry data set? And if you have continued to follow patients, is there anything even qualitatively you can share about how these patients are doing, or how disease has progressed within them relative to natural history. And then I guess briefly a second question. You talked about 5 to 10% of STD patients having GRN mutations. If you were to guess, on which end of that range do you think most of the evidence actually supports, and how confident would you be in driving patient identification efforts? You talk about misdiagnosis in this patient population. So anything you can talk about that would be great to hear. Thanks so much.
Great. Perhaps, Giacomo, you want to address the first question?
Sure. As you mentioned, the phase 2 study had 12 patients treated with latozinemab open label, and we had 10 matched controls from GEMFI at 1 year. The sample size is very small, and there are not many data at 2 years in the registries to support any kind of analysis as we did at 1 year. We have looked at biomarkers after 2 years. The sample size is small because there are not all the subjects who provide data at 1 year were available for 2-year follow-up. This is a very severely progressive disease. But what I can tell you is that the drug appears to be safe, well tolerated. The biomarker effect that we saw at 2 years in a smaller number of subjects are consistent with what we have shown at 48 weeks. And by mid Q4 this year, we will present the results in a significant number of patients, 103 symptomatic patients treated for 96 weeks. So we think this data will be the one that will be the most informative.
Anand, do you want to add anything?
Yeah, I can address the
second.
Second, what's the second question? You can.
I think the second question was around the FTD range, the 5 to 10%.
Yes, the prevalence.
The prevalence. Chris, would you mind re-asking that question?
Yeah.
What specifically?
Yes, where it is on the range.
Okay, where it is on the range.
So the fact is we don't exactly know, but as Sarah showed you, up to 40% of progranulin mutation carriers are misdiagnosed as other types of neurodegeneration. So you think there is a fairly high level of misdiagnosis of progranulin mutation carriers that sort of don't fall into FTD. There was a recent Los Angeles Times article on FTD that sort of put FTD at a very wide range of somewhere between 50 and 250 people, 1,000 patients. So that's something we think that what we are looking at Medicare sort of claims, and so we will have a much better idea on the prevalence. We will disclose at a later stage, but in general, again, as Sarah said, that once there is genetic testing and available, and once there is a drug, I think that the real prevalence will really come out.
That's helpful. Thank you.
Thank you so much. One moment for our next question. It comes from the line of Alex Stranahan with Bank of America. Please proceed.
Hey guys, thanks for hosting the R&D call today and for taking our questions. Maybe first on AL137 versus 037. You mentioned the reduction in reticulocytes between these two assets. Following up on a previous question that was asked, is this also a driver for which asset you'll take forward? And I guess did reticulocyte levels recover in your preclinical models?
Yeah, in general, safety is a parameter. What sort of Roche have shown with trontinemab is that sort of anemia is less of an issue than was initially thought. It's still, I think, up to 10% of the patients suffer from some level of anemia. In our non-human primates, yes, reticulocytes are very sensitive to both multiple blood draws and to transferring dependent drugs. So there was a reduction in reticulocytes, but there was a rapid recovery, and the real measure will be like chronic number of red blood cells and hemoglobin, and so far we don't see changes in these. But absolutely the safety will be a component of drug selection.
Okay, that makes sense. And then, how do you think about 001 and 101 as monoclonal antibodies versus other MOAs such as gene therapies in development for FTD, GRN, and maybe Alzheimer's as well? Obviously noting that the others are a bit earlier in development. Thanks.
Yes,
Arnon, do you want it?
Yes, I can take this. So, as you said, both sort of gene therapy, there's small molecules, there is enzyme replacement therapy, they are both very early, so we are comparing some hypothetical potential to an actual, like we are going to get pivotal phase 3 data in a few weeks. So it's really hard to compare, but conceptually, sort of the two gene therapy companies are struggling. One of them could not show sustainable elevation of progranulin. Sort of basically progranulin was elevated for a few months and then sort of went down. The second company had sort of significant AAV-related adverse effects, and again has to use immunosuppressive drugs and to significantly reduce the dose. And so there are technical issues with gene therapy. Also, gene therapy still infects a small percentage of the nerve cells, and you assume that this small percentage of nerve cells will be a depot, like will produce large amount of progranulin that will then diffuse throughout the brain for the other brain region. It's not clear how effective this is. And again, there will be a large disparity between some regions of the brain that have very high level of progranulin, other regions of the brain that have maybe insufficient level of progranulin. It's not going to be a homogeneous distribution, and we offer the correct level of elevation. We offer homogeneous brain distribution, so we think conceptually at least that our drugs could be superior. So the two gene therapies, again, have safety issues, I think have durability issues, and I think we also could have efficacy issues that they didn't really show any benefits outside of again, transient progranulin elevation. The enzyme replacement therapy is still early. It was on hold for several months because of adverse effects. It's requiring, again, immunosuppressive drugs to deliver it. It's not clear how viable it will be. And again, it will be a big discrepancy between very high level of progranulin in the periphery versus we'll see if sufficient level of progranulin in the brain. And there are also small molecule drugs that try to activate transcription from the progranulin gene. In the past, sort of these small molecules were tested and failed. And there is a small molecule drug that sort of blocks sortilin, does conceptually what we do with an antibody. And again, it's very early, so you don't really know the off-target activity. Progranulin sortilin is a part of a very large family of sorting receptors with very similar structure. So it's not clear what the off-target risk or adverse effects of a small molecule will be, what the durability will be, and what the ultimate efficacy will be. So we think that at this point we are in a very different place from competitors, both with how advanced we are and the scientific rationale for the program.
Got it. Thanks, Arnon. Appreciate the color.
Thank you so much. Our next question is from Greg Suvanave with Mizuho. Please proceed.
Hey, good afternoon or good morning where you guys are. Thanks for taking my questions and thanks for the presentation. I just wanted to revisit INFRONT-3 real fast, just on the co-primary endpoints. Could you just remind us if you need to hit a p-value that's 0.025, or is there room to hit 0.05 on both? I assume the former, not the latter. And as a follow-up, if you do end up seeing positive data, what are the gating factors in terms of next steps to be able to file the drug? If you could just remind us whether there are any CMC-related issues. Obviously you'd have to meet with the FDA, but just trying to get a sense of how, quote-unquote, quickly you could actually indeed file for approval. Thanks.
Maybe Giacomo can address the first question, and I can take the second part.
Sure. So we don't split the alpha. For the trial to be positive, both co-primary endpoints need to be met, and they are tested at the same time, not sequentially. So both the CDR plus NAC as well as plasma progranulin, they need to show a p-value that is p less than 0.05.
I think in terms of just the BLA, etc., if the data are positive, we are already poised to be able to start working on getting the BLA filing going, and we are on track for any of the commercial manufacturing activities to do that in a rapid manner.
Okay, thank you for the answers to both those questions. And just one last follow-up. With regards to any potential next opportunities with latozinemab, would you think about potential label expansion besides just potentially the asymptomatic patients? I know a long time ago there were interests in other indications as well for AL001.
Giacomo?
We are currently having those discussions both internally and with our partner GSK. So this is a very timely topic, and we'll disclose further details in a future date.
Okay, thank you very much.
Thank you so much. And we have a question coming from the line of Pete Stavropoulos with Cantor Fitzgerald. Please proceed.
Thank you for taking my follow-up. A question on the scales to be used in INFRONT-3. For FRS, is this a more or less sensitive measure, compared to CDR, some of the boxes, especially for certain, let's say, where you are within a disease stage, early versus later? Will we be able to see different rates of improvement on FRS versus CDR over the 96 weeks of treatment? And is one better for asymptomatic versus symptomatic? How should we be thinking about that when the data comes?
Giacomo?
Yeah, great question. So most of the data that we have around the natural history, natural progression of the disease are the ones generated using the CDR plus NAC. The FRS, obviously, it's a scale of interest, is an exploratory endpoint. The sensitivity and rate of progression on the FRS are not very well known or described. So hence the decision to use the CDR as primary outcome measure, which is being validated in FTD as a whole and is the one that is recommended by the FDA. Will be interesting to see the results on the FRS, which is an exploratory outcome measure. And we'll also, obviously, as you said, investigate the effect, the change over time in pre-symptomatic as well as symptomatic subjects and the sensitivity to change over time. So it will be an important analysis.
Okay, thank you for that, and thank you for taking my follow-up.
Thank you so much. And now I will transition back to Katie Hogan for any webcast questions on the other side. Back to you, Katie.
Great. Thank you. I think we have time for one more question that we received online, and this question comes from Sean at Morgan Stanley. And Giacomo, this question's for you. What are the key differentiators of latozinemab compared to other therapies in development for frontal temporal dementia due to a granulin gene mutation? And how do you see its potential impact on the standard of care? And we're specifically talking about Prevail Therapeutics and Passage Bio.
Thank you, Katie. I think Arnon already addressed these questions, but I would like to highlight the fact that there are lots of unknowns around gene therapy. What we know is the safety profile that doesn't look ideal. One company has reported only transient increase in progranulin levels in blood, and in most patients, not all patients, which is very different from what we observe. And then the other point is the rationale that 50% decrease of progranulin levels are enough to elicit the disease frontotemporal dementia in mutation carriers, and with our approach we are able to restore progranulin levels to normal levels, while it's a little bit more unclear the rationale around the need to elevate several-fold higher than the normal levels. Then going back to the safety, the one company showed increase in NFL that reflects dorsal root ganglia toxicity. Then the other company had immune adverse reaction. So there is quite some safety liability for the use of gene therapy currently, and that pertains around progranulin treatment, but also other forms of gene therapy.
Great. Thank you, Giacomo. That's our final question for today. And operator, I'll turn it back over to you.
Thank you so much. And as I was no showing questions on this end, I will conclude the conference and thank everyone for participating. You may now disconnect.